వాడు ఎరకలోడు కావాలన్నాడు పందులకు. ఏమంటావు? ఓ టైగర్. Good afternoon. Good afternoon. Good afternoon, everybody. Okay. Today we discuss the sexual incompatibility. So here, these plants are the very best source for the food for human and animals. So, however, nearly around the 30,000 of the plant species occur on the earth. So here, out of here, 40 species provide the majority of the calories and protein to the human beings. So in 19th century, the sufficient knowledge of the inbreeding and outbreeding of hybrid vigorous and inbreeding depression of the possibility of the proceeding producing F1 hybrid seeds was the assembly. So here the Smith is defined the plant breeding as the art and science of the improving the genetic plants in the relation to their economic use. The plant breeding is an unique to science as the information it impacts such as the crop varieties, composite synthetic population is the important for the understanding the behavior of the plant. So here we have the appropriate and adequate knowledge on the uh, about the following necessities are there. So what are there? These are the genetic and cytogenetic principle for the proper handling of the material. The second one is the characteristic of crop plants for appropriate genetic manipulation and mode of the reproduction, whether which is the asexual reproduction or the sexual reproduction. So here, nature of the breeding. So another one is the floral biology, that is the time of the anthesis, duration of the stigmatic receptacle, the flower structure. So another one, the geographical distribution of the wild relative and ability of the germplasm. So here, the croplands may are very greatly with their respect their mode of the reproduction and pollination. So these two basic features are associated with the precipitation of the generation. So based on this reproductive system, plants are categorized into three types are there. The first one is the sexually propagated plants. Second one is the asexually propagated plants. The third one is combined, that is the vegetative and combined means vegetatively and seeded propagated plants. So here the sexual propagated plants and vegetatively seeded propagated plants distinctly in the mode of the pollination. This can be grouped as the self-pollinated and cross-pollinated. Often another one is the often cross-pollinated plants with the 10 to 10% of the pollination and 90% of the self-pollinations are there. Next one is the factors that is the regulate the recombination in the plant. So here, so there are the five factors that regulate the recombination in the plant. So these, what are the five here? Entomological. In entomophilus, what is the entomological characters here? The entomophilus plants variation in the number kinds of the activity of the insect pollen vector may bring about the alternation in the breeding system. The vectors may be affected by changes in the environmental factors such as the light, temperature, humidity, rainfall, and predators. So the second one is the geographical and climatic factors. There is a variation in the breeding habitat from one geographical area to another, such as the temperature, humidity, precipitation, interaction with the other organisms. Okay. So this is the third one is the genetic factors. So here genetic factors in Lycoppersicum esculentum here dedicate the arrangement of the anther in the tube around the flowers is controlled by a single gene. The anthers are held by hairs and affected in to promote the selfie. Okay, the selfie. So this is the next one is the fourth one is the experimental modification. So self-incompatibility may be circum circumvented by the various such as means the complete outcrossing may be achieved in the monoecious plants by treatment of these seedlings with the male hybrid, which cause the inhibition of the staminated flowers. The monacium of the GMS may be readily already by the number of environmental factors including the photoperiod, temperature, soil conditions. So for fifth one is the selections. Selection factor is also very important in the recombination regulation. So long distance dispersal here. Long distance dispersal feature is the in Plumbaginacea family. Okay. The Flambagenaceae family, the floral dimorphism is found in the group near the distribution center, and the member are outer breeds. In areas far from the distribution, the central self-fertilization species are found. In the breeding system and habit, another feature, which is the self-incompatibility, annuals are the relatively, which is very rare condition. Okay. So in onions, is the normally cross-pollinated species that is the unique tolerant of the inbreeding. So in this species, some variety shows on the reduction vigorous on the continuous selfing, whereas other shows slight to moderate inbreeding depressions are there. So here, what happened here? The unfavorable genes are segregated on inbreeding and upon fixation brought about by the homozygosity produce the lines that is the possess the different genes are there. Different genes are gene complexes. So in other species that is reproduced by the self-fertilization homozygosity in the normal situation for most species are there. So here recessive deleterious mutations became the homozygous soon after their origin and the eliminated promptly. So in F1 hybrids between the lanes are fully vigorous and display heterosis. The values of the inbreeds rest on its ability to produce superior hybrids in combination with other inbreeds. The improvement of the establishment inbreeds had one or more of the following objectives are there, that is the to increase the productivity of the inbreed themselves to facilitate the production of the hybrid seed. The second, to fix up the inbreeds, so they are to produce hybrids to improve in the disease and insect resistance standing ability of the specific inbreed, so as to increase the yielding ability of their hybrids. So here self incompatibility. What is the here self incompatibility? In nature, there is the different floral adaptations have been evolved to prevent the self pollination, to stop the self pollination. Okay. So so what happened here? The self incompatibility has been reported in 66 family. So these morphologically self incompatibility are the two types are there. So first one is the heteromorphic incompatibility. Heteromorphic self incompatibility. It is the characterized by the occurrence of two or three, that is morphologically distinct mating the types within the species, which can be easily recognized without the breeding test. So the difference in the mating types are generally with the respect to the relative length of the stamens and styles. So this is the system to incompatibility reaction may be controlled by one gene with two alleles. There is the dimorphic species are by two genes with the alleles with the tristle. Other features, the heteromorphic incompatibilities are there. So that is the incompatibility react with the determined the sporophytically. The growth of the incompatibility of pollen tube is inhibited in the style. Dominance between the alleles of the incompatibility genes is expressed both the pollen and style. So next one is the homomorphic character. In this category, all the mating types with the species are morphologically distinguishable and require proper breeding test for their recognition. So here, depending on the origin of the factor, depending on the mating types or the pollen size, two categories are self-incompatibility are there. That is the geophytic self-incompatibility means gametophytic self-incompatibility. Another one is the sporophytic self-incompatibility. Gametophytic self-incompatibility and the sporophytic self-incompatibility. In gametophytic self-incompatibility, the incompatibility process determined by the genotype of male gametophyte. It is the example of the Poaceae, Solanaceae, Iliaceae members. So another feature is the sporophytic self-incompatibility. The incompatibility process is controlled by the genotype of the sporophyte tissue of the plant, which are the pollen is derived. That is the Asteraceae and Brassicaceae. Here, the mechanism of the self-incompatibility is the quite complex and is poorly understood. The various phenomena observed in the self-incompatibility mating the groups into three categories are there. The first one is the pollen stigma interaction, pollen tube style interaction, and third one is the pollen tube ovule interaction. This is the self-incompatibility. The first one is the pollen stigma interaction. We already discussed yesterday discussed that one. Pollen stigma interaction means this interaction occurs just after the pollen grain reach the stigma and generally prevent the pollen germination. So here what happened here? Only the specific gene, specific pollen grains, they are to reach the stigma, they are to identify by the stigma, they will be germinated. But here the structure of the stigmatic surface appear to be involved in the difference between the two systems. In the gametophytic system, the stigma surface is plumose having the elongated and receptive cells and known as the wet stigma. And the incompatibility pollen grains generally germinate on the reaching the stigma. The incompatibility react occurs at the lateral stage. There are the clear-cut serological difference among the pollen grains with the sporophytic type. In the sporophytic system, the stigma is dry and covered with a hydrate layer proteins known as the pellicle is present. There is evidence that the pellicle is involved in the incompatibility reaction. So here the sticking is difference in the stigma antigens related to the sporophytic allele composition. So after pollination, within a few minutes of the reaching the stigmatic surface, the pollen release the exudate with the either proteins or glycoproteins. These exudates include the immediate callus formation in the papillae. So often the callus is deposited on the young, producing the pollen tube to prevent the any further germination of the pollen. This is the sporophytic system. The stigma is site of the incompatibility reaction of the pollen tube across the stigmatic barrier. So here there is no further inhibition of the pollen tube growth. In the homosporic sporophyte system, the incompatibility reaction of the pollen is probably due to the deposition of some compounds from the anther and the pollen enzyme. Okay, so here this is the pollen stigma interaction. In the so second one is the so this is the this is the reversal. So here pollen stigma interaction. This is next one is the pollen tube child interaction. Pollen tube style interaction. In the most cases of gametophytic system, the pollen grains germinate and pollen tube penetrate the stigmatic surface, but in incompatibility combines and grow the pollen tube to related with the stigma. In the Inotera, in the latter cases, the protein polysaccharides in this pollen tube, which leads to the degeneration of the pollen tube. So next one, the pollen tube ovule interaction. In some cases of the Theobroma and Coco, the pollen tube to reach the ovule and effect in the fertilization. So finally, the incompatibility combines the embryo degradation at least and the early stage of the development. So here some incompatibility. Incompatibility here mechanisms are there. So here incompatibility mechanisms means so here what is the incompatibility mechanism? In nature, the different floral adaptations such as the dichogamy or heterogamy and unisexuality. So dichogamy, heterogamy, and the unisexuality have evolved to prevent the self-pollination, but the most widespread and effective natural device to enforce outbreeding in self-incompatibility. So here protandry. Protandry means protos means first, andros means male. So here protos means first protandry means male reproductive organs developed early than compared to female organs. That is when the anthers mature earlier than the stigma of the same flower. So anthers are mature earlier than the ovules. Here the pollen grains are carried down to the stigma of the flower. That is the cotton, for example, cotton and rose flower. That is the protandry. The next one is the protogamy. Protogamy, if the stigma here lost its the receptivity, recepticity by the time to anther dehiscence is called protogamy. So means here the stigma, stigma is mature than the ovules, means anther. This is the protogamy. So another adaptation here, unisexuality. The unisexuality means unisexual or decline is the condition in the flowers are either stamens or pistils. So any other is the decline means Another adaptation here, dioecium. Dioecium means the male and female flowers present on the different plants. The plants in such species are either male and female, that is the papaya, and date and that is same plant in asparagus also. In generally, sex is the growth by the single gene in asparagus and papaya. In some cases, there are the hermaphrodite flowers in addition to the male and females, and number of intermediate forms may also occur. So, establishment of the inbreeding here. In inbreeding has been associated with the unfavorable biological effect. This is the tendency arose in the accompanied by a small size and lensed vigorous reduced okay, reduced security and increase the number of defective and general weakness. So some advantages. What are the advantages are in biological significance of the sex incompatibility? So here we can see the sex incompatibility. We here so so heterostyle. So what are the advantages and biological significance of the sex incompatibility? What are there here? Okay. In the main advantage of the self incompatibility is to ensure the outbreeding, so as to maintain the heterozygosity. A large number of flowering plants are outbreeders, which means they are successfully fertilized only by the pollen of other plants and by their own. So in nature, a balance in breeding and outbreeding of the plants is regulated by intraspecific and interspecific incompatibility. So extensively, selfing of the plants leads to the highly homozygous individuals which have a very low survival value. So on the other hand, the intraspecific incompatibility brings out about the reproductive isolation and thus the responsible for the emergence of a new races and the species through the process of mutation and open hybridization. In free flow of the genes between two populations of the species, staff they are to regarded as two variant, and if the differences in the population are fairly large, they are the considered to the two distant races and species. Despite it is the natural value, sexual incompatibility may turn out be a seried handle in the plant improvement programs. For the several plants, and the self-pollination continues to be an important approach, which is to achieve the. In this context, self-incompatibility is a serious problem. Equally frustrating may be interspecific incompatibility because which is prevent a distant hybridization and thus the synthesis of better types by combining a good characteristic from different plants. The knowledge of these mechanisms controlling the incompatibility should be helpful in involving the effective methods to overcome it. So some methods are there to overcome the self-incompatibility. So what are the methods here which are the overcome the self-incompatibility? So what are the overcome methods which are self-incompatibility methods are there? So here for using the hybrid vigorous as a method for improvement the homozygous true breeding cultivars are extremely important. So obtain such the cultivars through the conventional method of the plant breeding, especially in the outbreeding crop, which is require many years of the patient work. Okay. It involves the selfing of many generations. Moreover, this is a technique cannot be applied to self-incompatibility plants. So before the introduction of the anther, the technique of selfing was on the chief method to obtain the homozygous individual. So here, for several plants where the anther culture has failed to yield the haploid and self-pollination continued to be an important approach to achieve. In this context, self-incompatibility is serious problem. Here, intraspecific incompatibility is the another major problem because it prevents the distant hybridization and thus the production of the better types by combining the good characteristics from different plants. There are many effective methods to overcome the intra and interspecific incompatibility. So first one is the bud pollination. So bud pollination is the most successful technique for the overcoming the self-incompatibility. It has been successful in both sporophytic and gametophytic plants. The systems such as the Brassica, Raphanus, and Nicotina, Petunia, the degrees of success varies from the species to species. The optional stage of the buds to for overcome the self-incompatibility ranges from 2 to 7 days before anthesis. It is likely that is the style, the factor complementary to the pollen factor for incompatibility reaction appears only at or just before the anthesis. Therefore, if the stigma is self-pollinated at bud stage where the factor not yet, pollen tubes will grow normally and affect the fertilization. Another one is the irradiation. So induction of the stimulation through the X-ray irradiation of the flower buds at the pollen mother cells was on the earlier successful technique for induction of the self-incompatibility. Lewis and Crowe worked on the extremely spontaneous and induced mutations at the locus in Inothera or Prunus. The subsequently mutations affecting actively of the allele in the both pollen and style have been reported in the trifolium. The breakdown of the intraspecific incompatibility following the irradiation is reported only in the gametophytic system. So another method to overcome the incompatibility, which is the stump pollination. Stump pollination means some investigations, some reporting have been attempt to started the distance to which is the pollen tube has to go in the pistil by removing the style pads partially or fully pollinating the cut end. This has been particularly useful in the cross involving the parents which is differ in the length of the style. In crosses involving the solanum, solanum penitentiatum and solanum canciforme and the Solanum penicitism, Solanum pinnati sectum, and bulboc castanum is remove the stigma together with a small portion of the style and curtain with a block of agar sucrose gelation gelation medium. The pollen grains were deposited on the medium and were covered with the pieces, piece mashed cotton wool. The hybrid seeds were successfully obtained in the both cases. Okay, so here, so and intravariant the intra intraovarian pollination. One of the incompatibility leaves on the stigma in this or in style. A direct introduction on the pollen suspension into the ovary would be helpful. This technique involves the injection of the pollen grains directly into the ovary and achieving the pollen germination. The pollen tube enter into the ovule, fertilization and seed development. Viable seeds followed the intra-ovarian pollination have been obtained in several members of the Papaver such as the Argemone mexicana and Argemone acrolenia, Papaver somniferum, and Papaver roses, etc. by the Kant and Maheshwari. So another method for the test tube fertilization, that is the in vitro fertilization, nothing but we call as the in vitro fertilization. So in this in vitro fertilization involves the culture of the pollen grains and ovules together on the suitable nutrient medium and which are achieving the germination pollen tube entry into the ovule and to form the double fertilization and development the mature seeds. So these pollen tube fertilization or in vitro germination or in vitro fertilization by the Kanto et al. In 1962 is reported successfully in the papaver somniferum by using this technique. So hot water and high temperature treatment. So this is also another method for the overcome the self incompatibility. So that the temperature significantly affected in the incompatibility reaction. In Inothera, the treatment of the style and stigma with hot water around the uh, around the 50 UV for the 5 minutes before the pollination, which is the inactivates the incompatibility reaction temporarily. It is also similar hot water treatment of either detached or intact styles at 50 UC for the 6 minutes before the pollination. So, which is the successful to overcome the self-incompatibility. The hot water treatment is considered to inactivate the protein which is involved in the self-incompatibility inhibition. The similar successful in overcoming the self-incompatibility by exposing the plants to high temperature has been reported for the Brassica rapana. Okay. The next one is the application of the growth substances and other chemicals also. We applied the some growth substances and chemicals. What are the application of the growth substances that has the naphthalostic acid and indole acid and gibberellic acid. This is one of the most common used method to overcome the self-incompatibility. This is a technique has been utilized to overcome the incompatibility in petunia and Tagetes, trifolium and brassica, lilium also. Generally, it is the presumed that the effect of the growth of substances is brought about by the inhibiting floral abstention and thus they allowing the slow growing pollen tube to reach the ovary. So here application of the 2,4-D, 2,4-D diclofenac acetic acid and over the 24 hours after the pollination was successfully achieving the intraspecific cross pollination in the solanum. We applied the 2,4-D, which is before the pollination, which is very successful. So use of the mixture of the naphthalene estamide and potassium gibberellate could be overcome the incompatibility in cross involving the Phascolarus vulgaris. Many chemical substances have been used to overcome the self-incompatibility. In Brassica olerans, the treatment with the stigma with the hexane before the pollination, which is very effective to overcome the self-incompatibility. The hexane has been suggested to remove or inactivate the incompatibility factor on the stigma. The hormone application is being very successful in many some gramineaceous members. So another method for the somatic hybridization. So here this is the isolating protoplast and culturing them into the complete plants to prove the useful in the overcoming all incompatibility barriers. In this method, the protoplast of the plants to be crossed can be isolated and fused with the protoplast can be cultured to obtain the complete plant. So here hybridization, somatic hybridization means to isolate the cytoplasm or protoplast. By using this technique, both intraspecific and intragenetic hybrids have been obtained. What is that one? Here, somatic hybridization. The first success in the somatic hybridization was reported by the Carlson et al. In the Nicotina longest fish and Nicotina glauca. Later on, several intraspecific hybrids have been obtained, which is sexually incompatibility. So this is the some methods to overcome the incompatibility, self-incompatibility. So here we discuss the incompatibility advantages and incompatibility mechanism. So some self-incompatibility adaptations. What is that one? Heteromorphic and okay, homomorphic conditions are dichogamy and hetero, dichogamy, heterogamy, and protandry, protogyny, and unisexuality. These are the some adaptations to the incompatibility. So here, the inability of the functional male and female gametes to effect on the population in particular combination known as sexual incompatibility. We discuss that one. What is the inability of the functional male and female gametes to effect the population that only called incompatibility. So in nature, the floral relations, the angiosperm, that one is the dichogamy, heterogamy, and unisexuality, which are to prevent the effect on the natural devices. So in self incompatibility, there's the pollen stigma interaction, pollen style interaction, and pollen tube interaction. So this is the incompatibility, self incompatibility. Okay. So tomorrow we discuss the endosperm. Okay. So okay, and thank you for attending the class. Hmm? Okay.